Hello. We are going to be covering fluid and electrolytes and acid-base balance, but I am going to break these up into two different videos. The notes will be the same, the PowerPoint will be the same, but I'm going to break it up because it's kind of long and it's first we have this one particular subject. Acid-base balance becomes another whole ball of wax, so to speak. So you'll find two different videos, Zoom videos for these two um, chapters. Okay. So I am sharing my screen and we'll start with fluid compartments. Intracellular fluid is the fluid that's found in all your body cells, and that constitutes about 2/3 of your total body water. And it makes sense because you have trillions and trillions of these cells, and that's where the majority of fluid will be found. About 1/3 is outside of that, and that's called extracellular fluid. Excuse me. And think about what I do is I think about um, the circulation of blood, and I kind of follow that. And so, okay, what would constitute extracellular fluid? Well, the plasma. Remember, 55% of blood is plasma, and 92%, 90 to 92% is water. And then as blood passes through the capillary bed, we lose water into the interstitial space. So right there. Then what does not get reabsorbed, so to speak, on the venous side of the capillary bed, osmotically drawn back in, is picked up by the lymphatic system. So it's in lymph. It's in cerebral spinal fluid because blood, is the source of production of cerebral spinal fluid. And then we have the serous fluid in all the different joints of the body, or let's put it, the synovial joints, serous fluid found between the serous membranes. We have different gastrointestinal secretions, saliva, tears, and so there's an abundance. Water is the main component. Males are about 60%, females are about 55%. Solutes, are all the different electrolytes, that can be found. And now we're at this point in A&P2, we're familiar with all these different electrolytes: sodium, potassium, magnesium, phosphate ions. We have bicarbonate ions, and so forth. Non-electrolytes are unable to ionize due to covalent bond structures, and those would be things like the organic molecules that we looked at in digestion in the chemistry lab in A&P1: glucose, amino acids, proteins, cholesterols. Fluid differences reflect the sodium potassium pump activities to maintain a resting membrane potential. Right. So remember that within the cell, we have high potassium levels, and outside the cell, we have low potassium levels. The highest amount of sodium chloride is found extracellularly, not intracellularly, and forever, because this will really help you understand why changing levels of potassium and calcium, can lead to cardiac irritability. So in extracellular fluid, we find high levels of sodium chloride. Inside the cell, we find potassium phosphate ions, because that's going to be a good buffer in there, and proteins, because a protein buffer is significant in the cell. So I kind of like this picture because it takes each electrolyte and compares and kind of contrasts where it's located either in blood and plasma or in intracellular fluid. So with very, very few exceptions, where you have something in high concentration in extracellular fluid, which would be blood plasma and interstitial fluid, the red and the blue regions, you would be low in intracellular fluid, and vice versa. So here, looking at sodium, it's very low inside the cell in gold, so you would expect the levels in the blue and red regions outside the cell to be very high. Potassium is going to be the opposite: very high inside the cell, low outside the cell. Right? Calcium is found almost, in smaller amounts, but outside the cell. Magnesium is higher inside the cell. I'm not so concerned about magnesium. Bicarbonate, we know that that's well, we will go on to learn when we look at acid-base balance, that bicarbonate is the primary buffer extracellularly. And so therefore it's low volume inside the cell or quantity. Chloride ions go with sodium, so high outside the cell versus inside the cell. Phosphates and proteins are going to be buffers within the cell, so they're going to be found more abundantly within the cell. And so I think it's kind of helpful to kind of look at that and at least understand that they're moving in opposition to one another. If you can't remember each one, then that's fine. But there's sort of a rationale behind it. You should know sodium chloride, way higher concentration outside the cell. Remember, that's why we have the sodium potassium pump to maintain the location of sodium potassium and therefore maintain appropriate resting membrane potentials. But in regular body cells, other than muscle cells and nerve cells, sodium chloride, where salt goes, water goes. Right? So it makes sense that you'd have more sodium chloride outside the cell because we want to regulate the size of the cell. If there's too much sodium chloride in the cell, then it's going to osmotically draw water into it, and the cell will expand and maybe lyse or rupture. Right? So sodium chloride always higher on concentration outside the cell. Okay, so fluid movement between the extracellular and intracellular regions. Exchange is regulated by osmotic pressure. Water moves freely. Solute movement depends on solute concentration. And so a change in extracellular fluid, and I'm separating here. Remember when we looked at hypertonic solutions and hypotonic solutions, the effects it had on red blood cells and AMP1, can lead to compensating changes intracellular. So when we looked at the effect of a red blood cell in a hypertonic solution versus a hypotonic solution and an isotonic solution, we knew that, if we had a cell in a hypertonic solution, which means we have less water outside and a high concentration of solute, then it draws water. Water will osmotically move from where it's in high concentration inside the cell out, and it'll cause the cell to shrink, right? So where salt goes, water goes. Right? Remember, we have aquaporins, and that even some of these small molecules can sometimes force their way through the phospholipids of the phospholipid bilayer. Hypotonic solutions would have the opposite effect. Now we have, a high concentration of water outside and a low concentration of water relatively inside the cell, so that the inside of a cell is acting like a hypertonic solution. So what does it do? It draws fluid, into the cell and causes it to lyse. And I just want to back up just for a second, because I've now said this a couple times, and sometimes I say this too many times, and then I say it backwards, so I think I just did. Right, so I just want to clarify. If the cell is in a hypertonic solution, the water is drawn out of the cell and causing it to shrink and crenate. I think I said lyse. I'm terribly sorry if I did. So in a hypertonic solution, it draws fluid out of the cell, causing it to shrink and crenate. And in a hypotonic solution, water floods into the cell and causes it to expand and lyse. I think I need another sip of Starbucks here. Okay, water balance. Now, having looked at the kidneys, we understand, that if we don't use hormones like antidiuretic aldosterone and renin, is that if we drink a lot of water, we're just going to produce a lot of urine. We don't reabsorb water, at the kidneys. With certain exceptions, without having these hormones present. And, there are certain exceptions. We know that the descending loop of Henle is permeable to water only, but otherwise to absorb water, actively, abundantly, we need to have those hormones. So essentially, the fluids we take in excess of being in our normal hydrated state is what we're going to lose, right? So the fluid we take in and is going to be the fluid that leaves the body in one way, shape, or form. So we're going to talk about that. So under normal hydrated conditions, so you're starting at a hydrated point. If I drink a quart of water, generally in the next 2 hours is going to produce about a quart of urine. All right. So what comes in is what's going to be lost. Thirst is triggered by a decrease in plasma volume or an increase in essentially the solutes, in plasma, the concentration, of the of blood. Antidiuretic is the major hormone that regulates water loss. All right, so if we're dehydrated and we're producing urine, we're going to secrete antidiuretic to try to reabsorb water alone, from at the kidney tubules. The stimulus for that can either be low blood volume, low blood pressure, or it could be an increase in that plasma osmolality solutes in blood, and that stimulates the hypothalamus to release, antidiuretic from the posterior pituitary. These are our general sources of water intake and loss. When it comes to average intake, and I don't ask you the average volumes, but if you can see where the majority of fluid is coming from, is coming mostly from the beverages, that you drink, on a regular basis during the day. Foods, because they're very hydrated, especially fishes and fruits and vegetables and meats. I mean, there's a lot of water in those substances. And then due to metabolic reactions, chemical reactions in the body. Where does it leave? Well, there's only so many ways out of the body anyway. So it's either going to be eliminated in urine, which constitutes the majority of it, but we also lose fluid in feces, in sweat. When we breathe in and out, we try to hydrate the air as we breathe it in, and we moisturize, so we moisturize it as we breathe in. And we know that when we exhale and you breathe on, let's say exhale on a mirror or on your glasses, we know we have condensation there. So when we respirate, it's a source of losing water as well. It's a water imbalance disorders. Well, dehydration, hypotonic hydration, and edema are examples. Dehydration is generally water loss from extracellular fluid that exceeds water intake. So essentially, you have not sustained anything extraordinary, other than you have not taken in fluids. Maybe you were outside working in the yard or at the beach all day, or taking the dog for a long walk, which hopefully you took water at least for the dog, and you did not take in any fluids. Then you are dehydrated. Your concentration of urine is very concentrated, and the concentration of your blood, that ratio of 55% plasma, 45% elements, is going to be thrown off. Water leaves the intracellular fluid, the cell, to maintain the osmolarity, and thus these cells may crenate. What are some conditions that may cause severe dehydration? Well, I just gave you a few. So when you're dehydrated like that, the blood, the plasma that the cells are sitting in, in blood, and thus this will translate to other body cells as well, is that those fluids are acting like a hypertonic solution. So it's going to draw fluid out of the cells. When you have severe dehydration, an application would be to just give an isotonic or maybe a lactate ringer kind of IV drip, because you just want to restore the fluidy compartment, of plasma. And so it's, once you just start an IV drip, you can rehydrate rather quickly. You'd always, if you're that dehydrated, you might want to wait before you take that, severely dehydrated, to take anything orally. But once that starts, then you can chew on ice chips and things like that that will slow down water absorption, in the stomach, or kind of keep it at an even pace. It's not generally a good idea to just then swallow and try to chug down a quart of water. So you sip and sip and sip and let your body slowly absorb all that water. Not so slow that, it takes 6 hours, but, you should, rehydrate over time, and rehydrating with, actually, if you can keep down food because you're not so severely maybe dehydrated, or, have any kind of heat issues going on, like heat stroke, is that eating some, just chewing a little bit of fruit, that's has a lot of water in it, like an orange, you know, vegetables that have a high amount of water, like celeries and things, you know, those are good sources of hydration. Anyway, moving on. Hypotonic hydration, exactly the opposite. You're overhydrated. You've taken in too high and rapid intake, high quantity, and a rapid intake of fluids. And that's why, you know, when you're dehydrated, this isn't really the issue to drink a quart of water. You know, there's other reflexive things that happen at the kidneys and things. So you just want to rehydrate more slowly. But hypotonic hydration means, you know, yes, you drank way too much water all at once, and your body starts to rapidly absorb those fluids. Well, now you've changed, you know, the ratio of plasma to formed elements in the opposite direction. You know, now you're so much more fluid filled. You are more, let's say, 80% plasma and 20% formed elements. And one of the risks that has is not only does it affect cell size, because now water floods into the cell and causes it to expand, you know, but you've also diluted all your electrolytes, you know, and when you severely dilute the electrolytes, then nerves and muscles can't actively, you know, open up these different voltage gates and allow these um, movements of sodium and potassium and things into and out of the cell to initiate different impulses and muscle contractions in the body. Remember, so you know, if it seems as if your sodium and chloride and potassium levels and calcium levels, you know, are really way lower than, you know, than the body can detect, and they can throw off the way the different cells that, you know, have voltage gates and things, and in the way that they're going to work. You know, if that makes sense. I'm not being very articulate about it. I'm sorry. You know, so the fact that you're diluting your electrolytes, you know, can cause you to have muscle and nervous issues. What effect does this have on the cells? It can cause the cells to increase in size, you know, and to lyse. Antidiuretic and any of the antidiuretic, aldosterone, and renin-angiotensin, you know, would be inhibited because you don't want to further reabsorb water at the kidneys. It may be that it becomes high enough where you start secreting atrial natriuretic peptide, you know, because blood pressure is going much higher, and we now need to actively block ADH, you know, aldosterone and renin from being secreted. And the way you would treat that is with a hypertonic saline, kind of an IV drip, where you want to draw as much, you know, fluid from the interstitial spaces, you know, into the blood and allow it to circulate to the kidneys so it can overflow. And the same sort of happens with edema. You would do the same thing. You use a hypertonic solution, and there's different grades of tonicity. You know, but the idea is that if you're hypertonic within the blood, then it will draw fluid from the interstitial spaces, and you know, when you have edema, because you have more fluid outside the cardiovascular system than you want, it will draw it back into the cardiovascular system, and then as it travels to the kidneys, you'll allow it to overflow. Now, using any kind of hypertonic solution and hypotonic solutions, you have to be really careful with those. They're not used that frequently, but there are occasions where you actually do. You just have to be careful, you know, especially if you have any risk of cerebral swelling. You know, if you have had any kind of injury or surgery, you know, brain surgery or anything to the brain, you know, is that you don't want to have a hypotonic solution that could cause cranial swelling, brain swelling of those of fluid compartments and things there. So that would be one of those issues. But on the most part, the hypertonic solutions are just going to draw fluid into the cardiovascular system. And as long as you don't have any kind of cardiac issues like congestive heart failure, is that it should be just fine. As you know, what we learned in the kidneys is as the blood passes through the kidneys, as long as you're not releasing antidiuretic and aldosterone, again, what the fluid that you took in should very quickly be produce urine and increase your urine output and produce a lot of dilute urine and be eliminated from the body. But in hypotonic hydration, it's so rapid that the kidneys are doing their job, but they just can't keep up with it. And you dilute everything, and your plasma and the cells, have a tendency to swell. In edema, that can occur for a number of reasons. I mean, if it's just a localized in your hand, it might be because, you just injured your hand and you have local inflammation. But when you have it kind of all over the body, it can be due to a number of things here that we can read about. Sometimes it's incompetent valves of the veins or along the lymphatic system. We have inappropriate amounts of albumin in plasma that maintain an osmotic gradient. And that could be due to liver disease. It could be due to renal disease where we're losing proteins, in urine. There's all kinds of reasons. It could be due to burns because we denatured lots of proteins. So you can read through some of these different etiologies. It could be due to longstanding hypertension where you've injured, let's say, the glomerular capillaries in the kidneys, and so we're losing proteins there. We have hypertension, so we're having damage at the systemic capillary levels, right? So there's all kinds of etiologies. And again, a hypertonic solution would be utilized, again, as long as we don't have any other kind of cardiac issues going on, it should be safe enough. You just have to monitor them. Right, so sodium and water balance, regulation of water output in urine is closely tied to sodium. And where sodium goes, chloride goes, and where salt goes, water goes. Right? Loss of water, decreases your blood volume and decreases blood pressure. Right, so if we're losing a lot of sodium chloride in urine, water is going to follow. We're going to decrease blood volume and blood pressure, and that may be on purpose. We may want to do that because the blood volume is too high. Blood volume is too high, and maybe the blood is kind of dilute like that, like hypotonic. The primary regulator of extracellular fluid volume and water distribution is sodium. Again, because where sodium goes, chloride goes, and where salt goes, water goes. It is the most abundant cation in extracellular fluid. It must be pumped out by active transport. Sodium is reabsorbed by the PCT and the nephron when necessary to maintain normal levels in extracellular fluid. We know that that's how aldosterone works. It actively reabsorbs sodium and secretes potassium. So as a quick synopsis, and now we've probably covered this 3 or 4 times, is looking at some of these hormones, that either affect blood volume and blood pressure and or just affect sodium. So although when we looked at increasing blood volume, blood pressure, we have antidiuretic aldosterone and renin-angiotensin. And when we have higher than normal blood pressure, sustained, then we're going to use atrial natriuretic peptide. And these 3 actually affect sodium levels. Antidiuretic affects blood volume and blood pressure, but it doesn't affect sodium. Remember, it's just for the reabsorption of water alone, at the kidney level. And so that's why it's not listed here. But if we wanted to group them together as what are the 3 that increase blood volume, blood pressure? Yes, we would have antidiuretic, aldosterone, and renin-angiotensin. And AMP, the third one, in blue here, is the one that works in opposition to those other 2 that would help to decrease blood volume and blood pressure. Okay, so aldosterone is released, sodium in the filtrate gets reabsorbed, we secrete potassium, you all know that. Renin-angiotensin, we know that renin is released by the juxtaglomerular cells of the afferent arteriole. It, is actively acts as an enzyme hormone, to convert angiotensinogen, angiotensin 1, and then angiotensin 1 needs to be converted to angiotensin 2 by ACE, and that, as a consequence of having the angiotensin 2, it is a vasoconstrictor, and it stimulates the further secretion of aldosterone and antidiuretic. So this, again, is just the same picture that I had from the textbook in the notes way back in endocrine. and also in the renal system, where we're looking at, how aldosterone works. Remember that aldosterone could be released because of high hyperkalemia and high potassium levels, but we're talking here about what are the three hormones that can affect sodium levels. And then here, and I probably should have put this in blue so that you would connect it to AMP like I just did, and I listed here as AMP in blue. So if you find that by the time you're reading this video, or you're listening to this video, I should say, and reading the notes, I may have changed this to blue just so you kind of color connect, like that. But if our blood volume is too high and blood pressure is too high, it's exerting a lot of pressure on the atria of the heart. And that's where atrial natriuretic peptide comes from. And so that will inhibit the first three, hormones being released and help to decrease blood volume and blood pressure. All right, so here it starts with the stretch of the atria. Again, you can follow this pathway, of how it's going to ultimately decrease blood volume and blood pressure. All right, chloride ions, it is the most abundant anion in extracellular fluid. Negative chloride ions follow potassium, because they're a cation. And chloride exchanges, remember, with bicarbonate to maintain proper a negative ion balance in red blood cells called the chloride shift. So all we're kind of coming back to here is that remember that chloride will also be exchanged with bicarbonate ions in red blood cells. If we're producing a lot of bicarbonate in the red blood cell, right, because there's a lot of carbon dioxide there, and that's pushing, the carbonic acid cycle reaction to the right, then as the bicarbonate builds up in the cell, it will leave the red blood cell and chloride will enter the red blood cell, to exchange and to balance these negative ions. If bicarbonate is really, really low in the red blood cell, then bicarbonate from extracellularly can diffuse, into the cell. Well, I don't think it diffuses, but it will enter the cell and chloride will leave the cell and go into the extracellular space. So they swap, and that's what we call the chloride shift. Okay, potassium is needed to maintain normal resting membrane potentials in nerves and muscles. Remember, aldosterone may be utilized when we have an excess amount of potassium, so that even though it will cause, at the kidney level to absorb sodium and chloride and water, maybe increase blood pressure, at least we're getting rid of potassium, because changing potassium levels, like this can produce a lot of cardiac irritability, and it affects the appearance of your EKGs. All right, so let's get into this part, importance of potassium. Remember that potassium should be high concentration inside the cell, and it affects the resting membrane potential in neurons and muscle cells, especially cardiac muscle, because cardiac muscle needs to be contracting appropriately all the time. So when we have an increase in extracellular, potassium, it's called hyperkalemia. And it causes a decreased resting membrane potential, which means it goes from, let's say, -70 towards 0, right? That's a decreasing RMP. It means it's becoming, it's going from -70 to maybe -60 to even threshold. And so it can cause depolarization prematurely, so to speak, and then it will be followed by a reduced excitability, so that it's going to be, so normally the potassium is supposed to diffuse from inside the cell, to the outside the cell when we depolarize. And then when we repolarize, potassium will flow during repolarization, after we've gone through depolarization. Depolarization is going to be the movement of sodium. And if we have a much higher concentration of potassium outside the cell, then our diffusional pathway isn't as extreme as it used to be, because you want it to be really high inside the cell and freely move to the extracellular space. Well, if we're really high potassium outside, that drive is not, that concentration gradient and that movement of ions isn't going to be as rapid, because the more severe the difference is in concentration of things, the faster things move. Right? So it ends up reducing your excitability. Right? When you have hyperkalemia, when you're looking at an EKG, you will have an elevated T wave. Remember that the T wave represents ventricular repolarization. And it's a small and rather small hump, upward deflection compared to the QRS complex. And so you will have a tendency, you will see a T wave that spikes upward. It's called an elevated T wave. When you have a decrease in extracellular potassium, it's called hypokalemia, and it causes hyperpolarization and non-responsiveness. So now the opposite will happen. Instead of the potassium, enter in, being able to kind of freely enter into the cell, causing it to become more positive and letting it approach threshold kind of prematurely. Now the opposite will happen. We actually lose more potassium from inside the cell to outside the cell, and that causes the inside the cell become more negative. So maybe going from -70 to -80 to -90. Now you're never, you're hyperpolarizing and never reaching threshold, and thus you have non-responsiveness. So what would that look like on an EKG? That will appear as an inverted T wave. Disruption in potassium hyper or hypokalemia in the heart can interfere with electrical conduction, clearly, leading to sudden death. Right. So potassium levels are criticals. When those go above and below normal, in blood specimens that sent down to the lab, you have to do critical alerts and start making adjustments quickly. And I believe something that would actually be worth noting is that you never IV push just potassium. You need to use other methods. Okay, bicarbonate, HCO3, it's the major buffer in extracellular fluid. And that's why we're going to be looking at this pretty extensively in the acid-base balance. You have the highest concentration in veins because we have the highest concentration of carbon dioxide there. Remember, in venous blood, we have high carbon dioxide levels. And knowing what we know about the carbonic acid cycle, just briefly, then we know the more carbon dioxide we have, the more hydrogen ion we produce. And thus the pH is decreasing. So we're going to have more bicarbonate available there to very quickly try to balance pH, and interact with those hydrogen ions as an immediate chemical buffer. Kidneys are the main regulator of reabsorption and secretion based on blood pH. Okay, calcium. Calcium is another one. Remember, we looked at cardiac muscle that we had the calcium plateau that's an integral part of a cardiac EKG. Or not really the EKG, but part of the action potential that you could draw out for cardiac muscle contraction. It's needed for strong bones and mineralization. It's needed for blood clotting, and it's needed for neuromuscular excitability. Remember that in muscle cells, we have the sarcoplasmic reticulum that stores and releases calcium ions. All right, and then as a brief review, parathyroid and parathyroid hormone and calcitonin, parathyroid hormone from the parathyroid gland and calcitonin from the thyroid gland, work in opposition to one another to balance blood calcium levels. When you have low blood calcium levels or lower than normal, we use parathyroid hormone. Remember, it has multiple mechanisms. It targets the kidneys to reabsorb calcium. It converts vitamin D to calcitriol. It stimulates the osteoclast to break down bone and release calcium. Calcitriol targets the small intestine to absorb calcium. And then when it's in excess, then we, higher than normal, then we use calcitonin. It decreases blood calcium levels by inhibiting osteoclasts and even stimulating osteoblasts to store calcium if there's any room in bone. And it allows calcium to just overflow into urine. Hypercalcemia, how will that appear on an EKG? You will have a shortened QT interval. And when you have hypocalcemia, you'll have a lengthened QT interval. Okay, so this is where I'm going to stop this Zoom and then I'm going to start another one because we're getting into acid-base balance, and that way you can, do this in portions, so to speak. All right, so I'm going to stop this and then I'll initiate a new one and it'll be a separate Zoom.